Thank you for the opportunity to speak at this conference. In fact, the first time I came to Poznan was in 1988 at the invitation of Dr. Andrei Maskevich and Professor Alex Goy, and I have been visiting Poznan multiple times. The title I put down initially was a very broad one, which is phase-separated biomolecular condensates in cell biology and virology. What I would like to do is make it narrower and talk about metastable condensates in interferon-induced antiviral MX proteins, and specifically talk about a paradigm shift in this area in the last 3 years. We've heard multiple presentations today of people treating cells with interferon or looking at tissues of patients with COVID-19 and finding increases in MXA in those tissues. My focus will be the MX proteins in the context of vesicular stomatitis virus, and towards the end, coronavirus too. I first talked about MXA condensates in person in Poznan in March of 2019, and it's been a fantastic time with this science since then. The concepts that I'll discuss are against a backdrop of changes in cell biology today. And these changes have to do with this concept of biomolecular condensates, which are phase-separated organizers of cellular biochemistry. I'm going to come back to this and define this better, but the thing you need to keep in mind is the concept of membraneless organelles. And these become involved in a variety of viral situations, the rabies, the measles, vesicular stomatitis virus, and it's now known the nucleocapsid protein of SARS makes beautiful droplets inside cells, and also the DNA viruses, adenovirus, Epstein-Barr virus, etc. My focus will be primarily the antiviral MX GTPases and the paradigm shift. Now, I know this is a celebration, so let's begin by celebrating RNA Goes Viral. When I first heard that title, I realized that viroids were discovered exactly 50 years ago, and the first paper was in August 1971. And this is a picture of Dr. Ted Diener. He's still alive and well at age 100. And clearly these viroids have ribosome-like properties. They are 300 nucleotides long. And when I went through grad school in virology at the Rockefeller in the early 70s, this was a spectacular achievement. Now, we've also heard of individuals or presenters at this conference talking about a nuclear event in the replication of influenza virus. It turns out that my mentor at the Rockefeller, Dr. Igor Tam, was the one, together with David Tyrrell, who's another giant in the interferon field, discovered that halogenated adenosine analogs, in this case, something called DRB. We all learned how to say this name in one breath, but I won't tax you with it. Back in 1954, showed that DRB inhibited flu virus replication. It then turned out that DRB inhibited cellular RNA synthesis. And from there came two lines of research. One, the fact that replication of influenza virus requires a nuclear RNA synthesis event, and that turns out to be 5' RNA cap snatching, which I know Powell Zamora has had some experience. Of course, it also turned out that this paper in 1960 was absolutely the first paper to show that RNA synthesis was involved in the life cycle of a DNA virus. This is with adenovirus. Now, I got involved a little bit later and made the discovery, together with Dr. Tom and Jim Darnell, that the way DRB works is by causing premature termination of transcripts. This turned out to be the discovery of RNA Pol2 transcription pausing. And today, I can now say And we can now say 4 decades later that 70% of metazoan transcriptional regulation takes place at the pol-2 pause site. This is about 100 nucleotides or 200 nucleotides downstream of RNA chain initiation. And this is inhibited by DRB, reversibly inhibited, because DRB inhibits the CDK9 enzyme. This specific event takes place in nanodroplets. I'm using a very important word right here, nanodroplets, which exist at both sites. And this was shown a couple of years ago by the MIT crew. There's an actual liquid droplet at the DRB pause site. And this is a very important concept in cell biology today. To back up a little bit, in 1957, we have Isaacson Lindemann discovering the interferon. And after this work was done in London, Dr. Lindemann went back to Zurich and did what everybody and their brother is doing right now in COVID-19, finding small animal models for influenza virus pneumonia. And in this case, they discovered that they had a strain of mice, the BALB-C in Zurich, inbred strain that was highly susceptible to intranasal administration of flu. It turned out that after a little bit of confusion, this turned out to be the mutant. And then the wild type strain, the MX plus, was resistant. So this is the origin of the discovery of the myxovirus resistance gene. And very quickly, Otto Haller and colleagues, also in Jean Lindemann, demonstrated in this paper in 1979 that this was because interferons, flu-induced interferons, cause an increase in MX1. Now, in the human system, we call it MXA. In the mouse, it's MXA and MXB. In the mouse, we call it MX1 and MX2. That's just the Otto Haller nomenclature. With interferon alpha, there is a marked increase, and several speakers today have talked about the marked increase in MXA upon interferon alpha treatment. But here's the point to make very clear. You see the MXA is in a structured, stringy appearance. We have a question about what that will be. Now, MXA is a 60 kilodalton dynamine family GTPase, and this has been studied extensively in cell-free systems. It makes large aggregates. It has a tendency to form dimers, tetramers, multimers, and so forth. So in the human case, we have MXA and MXB. MXA makes cytoplasmic structures. It's antiviral against flu A and VSV. MXB, the full-length MXB, sits at the cytoplasmic face of nuclear pores. It's antiviral against HIV, not flu or VSV. The mouse MX1, surprise, surprise, this is the autolog of MXA, is actually in the nucleus. This is antiviral against flu, but not VSV. And the mouse MX2 is in cytoplasm, is antiviral against VSV, but not flu. So you're immediately getting the point that the location of MX proteins in the cell affects what the protein does. This is what a GFP tagged MXA looks in different cells. I know this MISH2H6 is Dr. Andrei Maskevich's favorite cell out in Poznan, the melanoma vaccine cell. And the MXA is cytoplasmic. The mouse MX1 is nuclear. This is an example where autologs between species can be in completely different places in the cell and actually have very different antiviral effects. Now, what are these structures? Well, Mark McNiven and Otto Haller, going back about 2 decades, made a conclusion that these structures represented association of MX proteins with the smooth endoplasmic reticulum. Well, we got involved with this in a very indirect way and realized that we were experts in dealing with the endoplasmic reticulum, and realized the red marker here is the ER, the green is MXA, and the bottom line is they don't match. They just flat out don't match. We then proceeded to do detailed work to identify exactly what these structures were. We did CLEM, correlated light and electron microscopy, identified the GFP structures, did thin section electron microscopy. Bottom line, there is no membrane. And as soon as you realize there is no membrane, you realize that these conclusions are completely wrong. And if you actually go back to these papers, you find out that, I'm sorry, these are high-profile scientists. There's just no data for identifying the association of MXA with the endoplasmic reticulum. Now, the best news I can give you already is today, Otto Haller and I remain the best of friends. The real punch for us was in June of 2018, when we were characterizing the GFP MXA structures, and we exposed cells. These are the HU87 cells that you've heard about earlier today, to ELB. ELB is erythrocyte lysis buffer. It's hypertonic buffer, and these structures completely disappeared in a matter of a minute. And then we switched it back to isotonic buffer. The structures reappeared. We could go back and forth multiple times, and it led to the realization that we were dealing with something completely new. And in July of 2018, I ran into the fact that there was an entire field of science that I had absolutely no idea about even existed. And that was phase-separated organizers of cellular biochemistry. That was the concept of biomolecular condensates and membraneless organelles. This is a completely new thing in cell biology, and it's really hot stuff. So these images from Otto Haller's paper that were interpreted to represent MXA associated with the endoplasmic reticulum turn out to be MXA condensates decorating intermediate filaments, not a membrane structure. These also turn out to exist not just by transfection, but by actually treating cells. This is now the A549 cell treated with interferon alpha, and you see these beautiful dots. And I can tell you that these happen to be biomolecular condensates. The first time I talked about these as membraneless organelles was showing this slide in Poznan in a long time ago, let's just put it that way. And here are these MXA granules in association with intermediate filaments. Okay, to give you a quick overview of the subject, here are the MXA structures. These are antiviral against flu and VSV. And in the mouse, here are the nucleus structures. These are also condensates, biomolecular condensates. And I still have to define that term for you in a moment. And these are antiviral against flu, which has that nucleus step in its virus replication in its life cycle, but is not antiviral against VSV. And then in HU87 cells, we discovered a subset of cells in which the MX1, this is now the murine MX1, was associated with intermediate filaments. And in this case, surprise, surprise, this now has an antiviral effect against VSV. Now, what on earth is this phase-separated condensate? On a foggy day, you see dew on a glass window. And this is dew on top of my car. The dew drops are liquid. This is a phase separation of water vapor between gas and liquid. And if you drop the temperature further, you can end up with icicles on a scaffold. You can end up with streaks of ice on a scaffold, and these are condensates. And now here we're talking about a gas phase, a liquid phase, and a solid phase. Very simply put, in terms of cell biology, here are the icicles, and here are the droplets of MXA along the intermediate filament. And then here is long stretches of icicles, and here are the condensates on the intermediate filament. The biology of biomolecular condensates and membraneless organelles today is massive. Here are RNA binding proteins, first TDP43. All of these make droplets. Here is a translation promoting granule. And for our purposes, the most important thing, of course, the MXA bodies. But for our purposes, ribosomes, viruses in the cytoplasm, these make liquid droplets. And the nuclear capsid protein of COV2. In the nucleus, of course, we all know about nucleolus as a membraneless organelle, and then there are a variety of other things. Specifically, transcription droplets, nuclear STAT3 bodies. Earlier today, you heard about STAT1 and STAT2 activated by interferon, causing increase in gene transcription. Well, that's all in nuclear bodies. Now, there are a variety of tests one can make to demonstrate the liquid nature of these structures. And since we're running out of time, I am going to just race forward and say that in the case of MXA, these have a gel-like quality. When you do FRAP, fluorescence recovery after photobleaching, they have the feeling of a piece of plastic. We're basically talking about a conglomeration of pieces of plastic inside a cell. They're metastable, they can change shape, and they can fuse. And this is something I do want to say, spend a moment on. If you poke holes in the plasma membrane, these structures disappear. This is just saponin. This is back to the Heisenberg principle of uncertainty. The act of observing an object changes the object. So when people ask me, how do you can you purify these condensates? And the answer is only a subset that's resistant to saponin. As soon as I break the cell, the stuff falls apart. These are metastable. They need an intact cell to preserve their structure. What about the antiviral activity? Well, in the case of VSV, the nuclear capsid protein of VSV associates with these droplets. Now remember, even in uninfected cells, this is now the GFP-MXA transfected cells, even in uninfected cell, the red that you're seeing are really accumulations of liquid, phase-separated nucleoplasm. And then we can go a step further and ask about a structure like this, and we end up with a description which says, when the MX1 is in the cytoplasm, this has an antiviral effect. Location of the MX protein determines antiviral properties. I'm not going to go through the full listing here, but let's just simply say all of these MX proteins, MXA, MXB, MX1, MX2, make biomolecular condensates, make membraneless structures. Okay, now comes COVID-19. We've heard about the interferon background with COVID-19. We've heard about MXA being induced by type 1 interferons, and we then ask, is MXA per se involved in the antiviral response to COVID-19? And the first question I had was, do MXA condensates crosstalk with the nuclear capsid condensates? These we can do in BSL-2 whether it has an effect on live virus is something we still have to set up. We're in the process of doing the BSL-3 paperwork. Now, of course, we can demonstrate that the nuclear capsid protein of coronavirus 2 makes liquid droplets. Here are the liquid droplets. They disassemble with hexanediol, which disassembles condensates. They fall apart when you expose cells to intracellular edema. And we can then ask, can we find cells where the MXA condensates and the nuclear capsid condensates overlap? And the short answer is we can find them coexisting in the same cell, but no, they do not overlap. They are different organelles. And at that point, the biggest question for us and our reviewers at the NIH has been, does MXA per se have an antiviral effect against coronavirus 2? We're in the process of, we have a BSL-3 facility here, and we're in the process of doing the biosafety paperwork to be able to do this particular live virus experiment. However, this is an open casting call. I have no ego. I'm willing to give the GFP MXA vector to anybody who wants to run this experiment and has a BSL-3 facility. I know that my time is up. I'm going to stop here with the following comment. This phase separation and membraneless organelles in the cell, in the cytoplasm, and their role in viral replication. Oh yes, even in influenza A virus, maturation in the cytoplasm are taking place in the context of liquid droplets. This is a completely new way of thinking in cell biology. I'm going to stop here. My time is up. I'll take questions.
Thank you. Thank you very much for the interesting presentation, and it's time for question. Pavel?
Hi, Pravin. Thank you very much for your talk. It's really fantastic. I could just watch at your picture from the microscopy for hours. But I need to ask for SARS-CoV-2. Do you think that there will be some colocalization of your MXA compartments with the compartments which are very specific for replication for SARS-CoV-2 and coronaviruses, like the ER to Golgi intracellular compartments or the replication compartments?
The short answer is yes. I have images in my microscope which show some cells showing clear-cut overlap with the nuclear capsid and MXA. However, the biggest part of the story is absence of overlap. But however, my study section tells me, forget all that, tell us whether MXA transfection per se, not interferon treatment, affects live virus replication. And without that, they're not willing to give me 1 penny more. So my focus right now, I appreciate the question. I know what I need to do. I have a confocal 50 feet from my office. The problem for me right now is doing the BSL-3 paperwork. And I'm willing to do that experiment with anybody. It's a very simple experiment.
Thank you.
Any more questions, comments? I don't see. Thank you so much for your presentation once again, and hopefully meet in Poznań once again.
Yes, that's up to Dr. Andrzej Maskiewicz.